In Chapter 10, we'll be discussing photosynthesis. Photosynthesis as a process is going to be anabolic, so it is going to be somewhat of an opposite process of what we looked at in Chapter 9. It will not only be anabolic, but keep in mind that anabolic processes are endergonic. So as an endergonic process, it is going to require some energy be available to make this process happen. So by definition, photosynthesis is a process that takes light energy or solar energy and converts that energy into a chemical form. Specifically, we're going to pack that energy into organic molecules, which are a form of chemical energy that many other organisms do utilize. Now if we go back to this diagram that we looked at the beginning of Chapter 9, this diagram shows us the carbon cycle. We've already discussed at this point cellular respiration, and that cellular respiration breaks organic molecules open and releases the energy from them. When it releases that energy from them, that energy is used to make some ATP. At the same time, when we complete cellular respiration, if we do completely release all of that available energy from the organic molecules, we do yield carbon dioxide as a waste product. Carbon dioxide is what we typically call inorganic carbon. So in this diagram, we've got the organic carbon on one side and we've got the inorganic carbon over here. And as humans or as animals, we're not capable of utilizing that inorganic carbon. What the top part of this diagram is going to do is it's going to be able to take the carbon dioxide or inorganic carbon and convert that back into a usable organic form. In order to do that, we will have to have some energy, and that energy is going to come from sunlight. If we describe a couple different types of nutrition that organisms can have, we end up with organisms that are called autotrophs and heterotrophs. As autotrophs, organisms are able to sustain themselves. What this means is that they're able to use energy from some source other than organic molecules. So this may be from the sun or from the oxidation of inorganic substances. And when they take that energy, they're able to make their own organic molecules. So these autotrophs are capable of converting inorganic substances into organic substances. Plants are a great example of that. We will focus specifically on plants in this chapter as autotrophs. Now as animals, we are definitely not autotrophs. That's why that carbon dioxide is a waste product to us. For heterotrophs to get their organic molecules, they must consume some other organism that has either produced those organic molecules or consumed another organism that has produced those molecules. So as humans, we get our organic food or organic molecules by consuming plants, or we eat plants that at one point have eaten animals. Now plants specifically will be what we call photoautotrophs. As photoautotrophs, not only are they able to synthesize their own organic carbon, but they will be using light energy to do that. Now plants are not the only photoautotrophs that we have, so we do want to point out that there are other ones out there in nature. A good example of that is a lot of the organisms that do fall in the protist kingdom. This would include some single-celled organisms like a paramecium or euglena, as well as some very large organisms like the algae that you find in the ocean that make up the giant kelp forest. There are a few prokaryotes as well that are capable of photosynthesizing, so those will be our cyanobacteria. We will be focusing on the plants, but we do always want to keep in mind that there are other organisms or groups of organisms that are capable of photosynthesis as well. Since we are focusing on the plants, the leaves will be the major site of photosynthesis inside the plant. If you take a cross section of a leaf is just packed full of cells that are specialized for the very role of photosynthesis. And the reason that they're so specialized for this role of photosynthesis is because they are packed full of chloroplast. The chloroplast, we did introduce their structure back in chapter 6, But if we point out the structures of the chloroplast again, we want to keep these in mind as we go throughout the process, because we will be talking about specific reactions taking place in specific locations of the chloroplast. The chloroplast is a structure that has 3 membranes. If we point out those membranes, we are going to have an outer membrane, we're going to have an inner membrane, and both that inner and outer membrane, they're very close to each other, but they are phospholipid bilayers, and we will have an intermembrane space between them. So that fluid-filled space between the two, which is right in here, that will be what we call our intermembrane space. Somewhat similar to the intermembrane space that we had in the mitochondria. Now our third membrane is going to be the membrane that surrounds these green discs, which are called thylakoids. So this will be what we call our thylakoid membrane. So we've got 3 membranes. We've already pointed out one of the fluid-filled spaces that we have inside the chloroplast, and that's going to be the intermembrane space. We also have the space that surrounds the thylakoids, and this is called the stroma. And additionally, we have the very inside of the thylakoids, which goes by a couple different terms. It can be called the thylakoid space, or sometimes it's referred to as the lumen. So we do want to be familiar with both of those terms because they are both quite common when we talk about chloroplast. Now if we look at the process of photosynthesis, here's photosynthesis. This equation, we're going to take some carbon dioxide plus some water plus some light energy, and we're going to yield glucose or organic carbon, oxygen gas, and water. First off, notice that we do have water on both sides of this equation. So if we simply reduce it down a little bit or subtract off the extra water, this will be what we're left with. This equation should look quite familiar to you because it is very similar to the respiration equation. If we compare it to the respiration equation, here's our respiration equation. In respiration, first off, we had energy on the product side. That's why respiration was an exergonic process. Additionally, respiration is going to start with an organic molecule, and throughout the process, that organic molecule is going to get oxidized. When it gets completely oxidized, we are left with that waste product, which is our carbon dioxide. Now in respiration, we go back and draw our energy diagram, similar to what we had back in chapter 9. We're going to start with an organic molecule which has tons of energy packed inside of it, and specifically we were interested in the electrons. So the electrons in the glucose were very high energy electrons. We gathered them up in the process of respiration, and we passed those electrons to water. When we pass those electrons, or more specifically to oxygen gas, when we pass those electrons to oxygen gas, which then turns into water, those electrons are going to experience quite a drop in energy. This is why this reaction had energy that was actually given off, and why we called it an exergonic process. That's the respiration part of it. If we compare this with what's going to happen in photosynthesis, we're going to start with electrons in water. So these will again be very low energy electrons. So here we've got our electrons in water. We're going to take those electrons and ultimately we are going to pass them to the carbon dioxide. When we do that, that carbon dioxide is going to become organic. So it will become electrons in an organic molecule or electrons in glucose. We'll notice here that these are going to go from low energy electrons up to high energy electrons. And the only way that that's going to happen is if we have some energy to actually pack into this process to make it take place. That's where the light is going to come into play with all of this. So we will be taking low energy electrons and we will be giving them some extra energy to make them high energy electrons, which is what will then go to the glucose or the organic molecule. Now we can reduce down this equation a little bit further. If we do reduce it down further, kind of factor out all of the 6's in the photosynthesis equation, We want to notice that we're going to take carbon dioxide plus water, very simple ingredients really, and we're going to utilize some light energy to produce generic carbohydrate. Now we're not going to go all the way to producing glucose. We will stop with a 3-carbon carbohydrate and then with some oxygen gas as well. Certainly that 3-carbon carbohydrate that we make can serve as a building material to make larger carbohydrates. We just will not be following it quite that far. If we look at the respiration equation again, we want to very much be aware of what's getting reduced and oxidized in this process. Respiration was a bunch of redox reactions. There were a lot of individual ones that took place. But ultimately, what is going to get reduced in this process is going to be the oxygen gas. When oxygen gas is reduced, it turns into water. What will be oxidized in the process is going to be our organic molecule, which is the glucose. Now when this gets oxidized, it's going to turn in then to carbon dioxide. We said it is going to be an exergonic process in the end, so we will be releasing some energy. That energy is used to make ATP. Keep in mind that if we can follow the hydrogens in the equation, we really ought to be able to follow the electrons in the equation, because as the electrons have a negative charge, the positively charged hydrogens will follow them around wherever they go. Now if we take another look at the photosynthesis equation, this is also a redox process. So we will be collecting energy, or collecting electrons, and then passing them to another molecule. What will be reduced in photosynthesis is now going to be carbon dioxide. So notice here how carbon dioxide, when we move to the product side, is going to gain some hydrogens. And then what will be oxidized in this process is going to be water. And once water is oxidized, it does turn into oxygen gas, which is a waste product in this particular process, but it is really quite a valuable waste product, because we are dependent on that oxygen gas to run our respiration process. Notice how photosynthesis and respiration have opposite flows of electrons. So in respiration, we are taking the electrons from the carbon and we are moving them to the oxygen. And in photosynthesis, we are taking them from the oxygen in the form of water and we're passing them to the carbon. So in photosynthesis, the chloroplast will be responsible for splitting open the water molecule and gathering the electrons from the oxygen. We will be gathering up those electrons and then incorporating those electrons into the sugar molecules. It is going to be a redox process. It is a reverse flow of electrons compared with respiration. We said in this case we're going to collect them from the oxygen and pass them to the carbon. And it is going to require energy because we will be taking those electrons from a low energy state to a high energy state. That's where the light is going to come into play. So without light energy, this process is not going to take place. If we look at an overview of photosynthesis, photosynthesis is going to take place entirely in the chloroplast. So in the chloroplast, we will start with reactions which are called light reactions, and those reactions are going to take place in the thylakoids. So the thylakoids will intercept the light that is coming into the leaf. They will be able to absorb that light energy, and they will have specific molecules that are responsible for absorbing or collecting that light energy. When that light energy is collected, we will have the splitting of the water molecule, or in other words, the oxidation of the water molecule, where it actually loses its electrons. So right off at the beginning of photosynthesis, we will have our oxygen gas produced as a byproduct. And then we will take those electrons, which are low energy electrons, we will energize them, and we will pass them to NADP+. NADP+ is very similar to NAD+, except that we've got the P on there, which stands for a phosphate group. When that NADP plus gathers those high energy electrons, it will turn into NADPH. NADPH will be performing a very similar role to what NADH performed. In other words, it is temporarily holding on to electrons. It will later pass them on, because ultimately in this process we're going to reduce the carbon dioxide. So those electrons will ultimately go to the carbon. Also in the light reactions, we will be using some light energy to make some ATP. The light reactions, their purpose or their goal is to provide the Calvin cycle, which is the second stage of photosynthesis, with whatever its energy needs are. And that is ATP, and it's also energized electrons in the form of NADPH. The Calvin cycle is where the carbon dioxide will actually get reduced. So that means that is where the sugar is going to get produced. And when we use the ATP and we use the electrons in NADPH, we will be returning those used up molecules back to the light reactions so that the ATP can be reassembled and the NADP plus can be reduced again or gather more electrons. So if we look at this in another way, the light reactions will occur in the thylakoids, or what we often call the grana. They will convert solar energy into chemical energy, and that will be in the form of ATP and NADPH. At the same time, they will certainly be splitting a water molecule because they will be oxidizing the water, and then they will release oxygen gas as a byproduct. The Calvin cycle will be the part of the the part of photosynthesis that is specifically required for making the sugar. The Calvin cycle a lot of times is called the dark reactions, and it's called the dark reactions just to play off of the name light reactions. This doesn't mean that it specifically occurs in the dark, but it does emphasize that this is not the part of photosynthesis that specifically requires light. It does require, however, the products of the light reactions. So in the Calvin cycle, it will form sugar from carbon dioxide using ATP for energy and NADPH for reducing. This Calvin cycle is going to occur in the stroma, so right outside of the thylakoids. So that's a big overview of photosynthesis. We want to now look at the details of photosynthesis, and we will start with the light reactions. Because we are talking about the light reactions, it will definitely be important to initially discuss light, because light energy is what's going to power this whole process for us. Light energy is a form of electromagnetic energy, and that means that this is energy that travels in waves. If we draw or look at a typical wavelength or wave of energy, that wave of energy is going to have wavelengths, and that goes from the top of one peak to the top of the next peak. That's a wavelength. You can imagine that we could take this wave that is drawn and we could stretch it out like a spring, and we would have longer wavelengths, or we could compress it together and we would have shorter wavelengths. The wavelengths of electromagnetic energy is what determines its properties. Now, while light is energy that travels in waves, it does have some particle-like properties to it. And when we talk about a little bit of light, we refer to that as a photon. So you do want to know what a photon is. It's a discrete or a small amount of light energy, and we definitely want to make sure that we're not confusing that with a proton. So if we look at all of the different electromagnetic energies, here we see them. The difference between them is really all about the wavelengths. So the numbers that are shown in this picture indicate the wavelengths, and here we're seeing them all expressed in some form of meters. So the N stands for nano. So over on this end we're looking at 0.01 nanometers, and nano is 10 to the minus 9. So on this side we are looking at very small, short wavelengths. If we look at the part that is expanded in the middle, this is what we typically call the visible spectrum. That's the light that we think of. And then way over on this end, we've got radio waves, and this is in kilometers, and kilo is 10 to the third. So now we're getting very large wavelengths. So if we go through and name these, this first one would be radioactivity, then we have X-rays, ultraviolet light, visible light, infrared, and then we finally have radio waves. One end of this spectrum is considered quite dangerous, so that would be this end over here. The other end is considered pretty harmless. The reason is all about the wavelengths. If you look here on the bottom, we do have an inverse relationship between the wavelength and the amount of energy that's actually packed into that type. So the shorter the wavelength, the higher the energy that's there. So when we look at something like radioactivity, that has an extremely short wavelength. That means that it is extremely high energy. That means that if that energy actually strikes a molecule or strikes a cell, there's so much energy packed in it that it can cause quite a lot of damage to the cell. On the other end of the spectrum, the radio waves are very, very long, and they are so long that there is so little energy packed into them. So if they strike something, they cannot do much of anything. They're not going to cause damage. We want to definitely understand that inverse relationship between wavelength and the amount of energy. If we just zoom in on the visible spectrum is what we're interested in this chapter. That is the energy that's going to be utilized for photosynthesis. And if you've ever taken a prism or seen a rainbow in the sky, we know that the light, as we think of it, can be separated out into different colors. So we have the visible spectrum representing the different colors of light. These colors of light have different wavelengths, as you can see on this picture here. The purples and blues have shorter wavelengths, and then the reds and oranges have longer wavelengths. That means that all colors of light are not going to have the same amount of energy packed inside of them because they do not have the same wavelengths. So some energy or some colors of light will be more valuable than other colors of light if we're thinking just actual quantity of energy that they have. Now we're going to utilize that visible spectrum for photosynthesis, but we will have specific molecules whose job is to collect that light. Now these molecules will be called pigments. Pigments are substances that selectively absorb visible light, and this means that they are capable of absorbing certain colors or certain wavelengths, and if they absorb specific wavelengths, those wavelengths or colors disappear because they are being absorbed. There will be other colors or wavelengths that they are not capable of absorbing, and those wavelengths will be reflected instead. If we look at the pigment being shown on this slide, we have a blue pigment, and the reason that it appears blue is because all of the colors of light will come in and strike that blue pigment, and that blue pigment will be able to absorb the green, the red, the yellow, the orange, all of those other colors. But the color that is not capable of absorbing is blue. So that means that blue bounces right off of the pigment molecule, and then that's what we see. We see blue because the blue is bouncing off. If you compare the color of a plant, plants are generally green or some kind of a greenish yellow. This is because the light is going to come into the plant, it's going to strike the pigments that happen to be in the thylakoids, and those pigments are capable of absorbing pretty much all colors with the exception of green. So that means that the green light will be transmitted or reflected from the chloroplast, and when we look at the plant, they appear green. That's not to say that there aren't various pigments inside the plant. We will actually have a whole collection of pigments inside the thylakoids themselves, but obviously we do have some parts of a plant that are not green in color. So you can have very colorful fruits, reds, oranges, all different colors. We can have different colored flowers. Those brightly colored areas of a plant, they will have their own assortment of pigments inside, and those pigments may be reflecting orange, or they may be reflecting purple for that matter. If we can see the color of a substance or of a plant organ for that matter, then we should be able to predict which wavelengths are being collected or absorbed, and which wavelengths are being reflected. If we take a look at what we typically call the absorption spectrum of the pigments involved in photosynthesis, this shows us some of the various pigments that you would find in the thylakoid, and we can compare how much they absorb of all of the different colors of light. If we look at chlorophyll A first, chlorophyll A is just one of the pigments inside the thylakoid, and chlorophyll A we can see absorbs very well here in the purple and then some of the darker blue, and then it starts to drop off, and we have a little hole here where we're not getting much absorption at all. Then we pick up a little bit, absorb a tiny amount, and then we have another peak over here on the red end. If you look at where the gap is, where chlorophyll A is not absorbing light, it's right here in the blue-green area. So if you notice the color of the line that's drawn for chlorophyll A, it is blue-green in color. So chlorophyll A is blue-green. If we compare that with chlorophyll B, its spectrum is shifted just a little bit over, so we are getting more absorption over here in the blue area, but then we have a drop off right here in the middle with the green and slightly yellow area. Then it picks up again over here on the red side of things. Notice the hole for chlorophyll B is going to be more where we have the greenish yellow. So chlorophyll B is typically considered a greenish yellow type of color. The carotenoids represents a whole group of pigment molecules. These as a whole absorb very well in the purples and the blues, and then they don't absorb well in the reds, oranges, and yellows. So here we have the line drawn as orange, but different carotenoids would have different specific colors that they appear. If we think about what's in the orange carrot, beta carotene is an example of a carotenoid, and that one is clearly orange. It gives the carrot its orange color. Another common example is lycopene. Lycopene is found in tomatoes, and it gives tomatoes their bright red color. So if you think about all of those brightly colored fruits and vegetables that are reds, yellows, and oranges, they do have a whole bunch of carotenoids inside of them that are giving them that specific color. When we look at all of these absorption spectrums kind of drawn on top of each other as they are right here, notice that we have this gap or hole right here in the middle. There are no pigments there that are capable of absorbing green. So this means that even though there are a lot of different pigments there, they will be reflecting green light. The carotenoids may not be absorbing red, but notice the chlorophylls are absorbing red. And so they're all kind of helping each other out to cover as much of the spectrum as they possibly can, but we still have that little hole in the middle where we're not absorbing green. If we compare the absorption spectrum with the action spectrum of photosynthesis, the action spectrum shows us how much photosynthesis we get per color of light. Now we're really only going to be able to utilize the colors of light that we have pigments available that are capable to absorb that color. If you look here where we have the purples and the blues, we have a whole lot of photosynthesis taking place, and this matches up with where we're getting the absorption of the purples and the blues. We have a hole here in the middle where we have the green light, and then we have another peak over here in the red-orange area of the spectrum. So notice how this action spectrum would be different if we only had one pigment inside of a plant. If we just had, for example, the carotenoids we would see the very first peak, but we would be missing this whole second peak. So plants have evolved to have lots of different pigments, and by having lots of different pigments, they're going to be very efficient compared to, say, only having one pigment available. So we're having lots of pigments to increase photosynthetic efficiency. If we look briefly at the structure of a chlorophyll pigment, this molecule, very large in structure, and a few things to point out about this one. First off, we have two chlorophylls that we've looked at so far. We have chlorophyll A and we have chlorophyll B. And we saw that they do have a different absorption spectrum. So chlorophyll A was a blue-green in color, and chlorophyll B was a yellow-green in color. The only difference between these two pigments is the little boxed in area that we see up here. That one little section is different, and it is enough to cause the absorption spectrums to shift. Another thing we want to point out is that there is a big hydrocarbon tail here in our chlorophyll. This will be important because our chlorophyll molecules will embed into the thylakoid membranes. Keep in mind that the thylakoid membranes are phospholipid bilayers, and as such, the inside of them would be hydrophobic, or we could say nonpolar, and only nonpolar hydrophobic things would be able to embed in that inside core of the membrane. We'll see the same thing if we look at structures of carotenoids as well. They're going to be highly hydrophobic, and in some cases they are entirely hydrocarbons. So if we look at how the pigment is actually going to function, the light will come into the leaf and hopefully strike one of these pigment molecules that happens to be in there. When it strikes a pigment molecule, if it is the right color, that pigment will absorb the light energy. When it absorbs the light energy, an electron specifically will absorb the energy. And an electron that absorbs that energy is going to be boosted up to a higher energy level. At this higher energy level, this electron, we say that it is excited, or we could say that it's very unstable. Keep in mind that stability is correlated with the amount of energy that something has. So the more energy the electron has, the more unstable it is going to be. Now that electron, because it is so unstable, it's not going to stay there in that excited state for very long. So if left for a little while, that electron will fall right back down to where it was previously, which is what we call the ground state. In the ground state, it will not have that extra amount of energy. If it returns to the ground state from the excited state, it will lose some energy in the process, and that energy will be lost in the form of heat and then oftentimes as fluorescence. Well, that is not what we want to happen in photosynthesis. In photosynthesis, we want to have an electron that is excited, very energetic, and then we want to be able to capture or utilize that electron while it is still in that excited state. So we will have molecules that their whole role in photosynthesis is to basically reach down and grab that electron as soon as it is boosted up to that excited state. That will prevent that electron from falling back down to the ground state and releasing the energy that was collected. Now, all colors of light are not worth the same amount of energy. Keep in mind they had different wavelengths, which meant that they did have different amounts of energy. So blue light, for example, has a shorter wavelength, and it would have more energy packed inside of it, and that means that it would boost an electron to a higher state. Red, on the other hand, has a longer wavelength, and it would not boost electrons quite as high. If we look at the efficiency of photosynthesis, just so we can compare it with respiration, keep in mind that respiration was a very efficient process. Respiration was 40% efficient, meaning that 40% of the energy that was released from the glucose molecule did get packed away in ATP molecules. In the case of photosynthesis, we are much less efficient. We will start with basically 100% solar energy coming into the leaf. Right away, 60% of that energy will be lost in the form of non-absorbed wavelengths. Another 8% will be lost because it is reflected or transmitted. We will have 8% again lost as heat, and then we will lose some in metabolism as well. So when all is said and done, we're going to get about 5% of the light energy packed away in the form of a carbohydrate. Now, while 5% sounds like pretty poor efficiency, and it is poor efficiency, plants are able to be this inefficient because the light energy is not really limited. So there is an abundant supply of light energy, and there has not been the pressure to adapt and create a much more efficient process with photosynthesis. So we do want to definitely understand that photosynthesis, although we are using lots of pigments to capture as much of the energy as we possibly can, we are nowhere near the efficiency as what we had in cellular respiration.